Hello, everyone. I'm so excited to be able to share what I've been working on over the summer with investigating parasitism on herbivory and chemical communication in an insect herbivore. Something to know first is, what are parasitoids and what do we know about them? Well, many of you may know about parasitism, which is normally when a parasite leeches off of their host. Parasitoids are organisms that lay or insert an egg or a live larvae inside, which eventually kills the host organism. And this may have implications for their insect behavior. So if we look at Hawaiian crickets, we see that in the presence of parasitoids, they're changing their chirping behavior, which can have implications for their mating calls and reproduction. But also, we can see a cordyceps fungus, which is shown on screen, that has completely taken over the beetle, which is really interesting, and that can show that parasitoids can have a really important factor on controlling insect herbivores in an ecosystem because they display similar dynamics to a predator-prey relationship. But what do we not know about parasitoids? One thing that we might want to look at is their effectiveness. How do we know that a parasitism is successful? How do we know whether or not it's reducing consumption or rate of consumption in a plant, or even if it's affecting the fitness of the host? But more specifically, how are parasitoids or parasitism impacting communication in an insect herbivore? Going back to our example with Hawaiian crickets, we saw that in the presence of a parasitoid, they were changing their behavior. But we do not know if this is because the cricket was parasitized and whether it was an internal mechanism that was changing that behavior. So this opens up a very broad question on how parasitism is changing insect behavior in an insect herbivore. And more specifically, I will be discussing today about how parasitism in selected hosts is changing feeding behavior and how parasitism in a selected host is changing chemical communication. Now, I'm extremely excited to introduce the striped cucumber beetle, which you can see on the screen, which is the main insect host of our system. They are common agricultural pests to the cucurbit plant family, which includes things like summer squash, desert zucchini, and New England pie, just to name a few of my favorites. But they also cause a huge problem on these same plants because they cause a lot of plant damage. As you can see from the striped cucumber beetles completely having a buffet on the middle of that plant. In addition, they use a pheromone called beta-lactone, which they are able to produce mainly by males for aggregations. And it calls in other striped cucumber beetles to say, hey, this is a really good spot to eat. And also, I need some female striped cucumber beetles to be able to mate. But luckily, they are highly parasitized by a natural enemy, C. setosa, which is a tachinid fly. Here we can see a C. setosa on one of our flag markers in the field. Now, what's really cool about this tachinid fly is what we know about it is it is the natural enemy of the striped cucumber beetle. And the striped cucumber beetle doesn't normally have a lot of natural enemies because it sequesters the secondary metabolite, or the cucurbitacin, in the plant, which makes them very bitter and definitely not tasty because I took too many in with an aspirator this summer. But what we also know is the life cycle of the C. setosa. So for a really good example, I want to use this striped cucumber beetle plush that we made over the summer. And so normally the tachinid fly will come in and oviposit, or lay a live larvae inside the striped cucumber beetle, and now it is parasitized. Over 8 to 10 days, this will develop into its 5th stage, which is a pupa, and completely burst the striped cucumber beetle. And now this remains in the husk of what once was the striped cucumber beetle. Now this parasitoid pupa will then take 10 to 12 days to evolve into an adult eclosed fly, which we know as C. setosa, or the tachinid fly. Now, knowing this, we wanted to start with our experimental collections of the striped cucumber beetle and the tachinid fly at 2 locations, which was Felons Family Farms in Geneva, New York, and West Haven Farms found in Ithaca, New York. To start our experimental design, we did a 2x1 factorial design for herbivory trials, where we took these wild-caught females that we caught from a variety of different species, including summer squash and desert zucchini, and we isolated them for around 6 to 10 days just to rear out any potential parasitism that could have been from the field. From there, we separated them into two different treatments, those being non-parasitized and parasitized. The parasitized treatment were exposed to a female C. setosa fly, and we watched them in lab be parasitized by ovipositing a live larvae. Meanwhile, the non-parasitized beetles were exposed to a male C. setosa fly, which we know cannot oviposit an egg. From there, they were placed in their experimental cups, which you can see on the left side of the screen, with a summer squash leaf. We weighed them every 2 to 3 days for measurements, and during that time, we imaged the leaves for the amount of consumption in millimeter squares utilizing ImageJ. From here, we can see the experimental setup on the left-hand side with a lot of different replicates. And over the entire treatment, we saw 96 different replicates. Up on the top, we can see the non-parasitized treatment, which had about 46 different successful non-parasitized beetles. But we did find that 6 of them were parasitized before we caught them, meaning that they passed the isolation period and we found them parasitized in the cups. And also, we found 9 of those that did die. Then in our parasitized treatment, we found 18 that were successfully parasitized, meaning they went from this happy little striped cucumber beetle to a husk of what once was the striped cucumber beetle. From there, we also had 2 that were parasitized prior, meaning that they were formed this pupa on day 2 or 4, which is too early to be considered parasitized by our lab. And lastly, we had 10 that died during the experiment. Now, what's really cool is this is one of the first ever videos of a C. setosa parasitizing a striped cucumber beetle in the lab. And so we can see it in slow-mo that the tachinid fly is going to pounce onto the striped cucumber beetle, and then its abdomen is going to stick its ovipositor inside of the striped cucumber beetle, laying in the egg. And that is how we knew that they were parasitized. Now, the second part is how did we quantify or collect vititalactone, which again is that pheromone, group-based pheromone that the striped cucumber beetles produced. And we caught wild-caught males because, again, they are primarily produced by males. And we isolated them again to rear out any potential parasitisms. And similarly to herbivory trials, we separated them into non-parasitized and parasitized groups. Then we ran a volatile collection assay, which is also known as a VOC, for 3 days, around days 3 to 6 of the parasitized life cycle, and the vititalactone was collected. So we can see on the left-hand side of the screen, we had an air source that was pushed through activated charcoal to clear it, and then a bubbler to humidify it and be easier to collect these pheromone compounds. The striped cucumber beetles, which were about 6 per tank, were feeding on a pumpkin, and the vititalactone was then collected from there. This whole entire process to quantify and collect and then use gas chromatography was headspace volatile collection. And so this offers a lot of cool opportunities to ask questions in chemical communication, which include, do parasitized beetles even produce vititalactone? And if a beetle is even parasitized, is there a difference in the amount of vititalactone that it's producing at a population level? So now we're going to go on to some of our really cool experimental results. So the first thing that we found was that parasitized beetles consumed less leaf tissue than non-parasitized beetles over 12 days. And we see that these results were significant. We saw that the non-parasitized beetles were eating around the 700 mark, while the parasitized beetles were eating around the 300 to 400 mark. But a different perspective on this graph is that it is also saying that non-parasitized beetles are consuming and causing more plant damage than parasitized beetles. And we have to remember that the parasitized beetles pupate or die by days 8 to 10. So it is a good thing to see that even though they're dying on 8 to 10, they are consuming less. So this is great for farmers and agricultural implications. Our second result was that parasitized beetles consumed leaf tissue at different rates than non-parasitized beetles. So again, going back to their dying at around 8 to 10 days, we see that the graph only ends at 6. And that's because at days 8 to 10, we see a large drop, and that's because they're parasitized or died. So we can really only analyze these first 6 days. And from that, we see that non-parasitized beetles, which are displayed in red, are consuming around an average of 150 to 160 millimeters squared per day, while non-parasitized beetles, or parasitized beetles, immediately after they are parasitized, quickly drop in their rate of consumption, which is very interesting, and slowly return to their non-parasitized counterparts in consumption rate. But an unexpected finding that we found was that with dealing with fecundity, or the ability to lay eggs or reproduce in the striped cucumber beetle and parasitized females, we found that between non-parasitized and successfully parasitized, which was, again, the parasitoid pupa emerged, we found that non-parasitized beetles were able to produce eggs as expected, but parasitized beetles were not able to produce eggs. And this was a significant result, explaining another mechanism on how parasitoids may actually impact internal mechanisms inside an insect host. And lastly, we found that parasitized beetles may not be able to call or produce this aggregation pheromone vitidilactone, or call less than non-parasitized beetles. And so we can see through red that non-parasitized beetles produced a ton of vitidilactone, while parasitized beetles produce very little. And although this is preliminary data, because we don't have enough replicates to confirm this, it is trending towards significance. But what's also interesting to see is that this vitidilactone production aligns with our rate consumption, where as the vitidilactone was not being produced by parasitized beetles, the rate was changing for parasitized beetles, meaning that they were going back to normal levels, and yet still they weren't producing this vitidilactone, which is very important and may provide an interesting mechanism on how parasitoids are impacting chemical communication. So just to review, how does parasitism in selected hosts change feeding behavior? Well, we did see that they fed differently between non-parasitized and parasitized beetles, but we did not see that they fed at the same rate. In addition, in terms of how they changed chemical communication, we saw that parasitized beetles could produce vitidilactone, but we also saw that there was a difference in the amount of vitidilactone they could produce at a population level. So this can have many implications, starting with in 1925, the very first experiments on the ecology of C. tetosa started. And since then, this is one of the first papers that actually explores the predator-prey dynamics of this ecology. It's also important to, again, mention that the striped cucumber beetle is a huge agricultural pest, common in Northeast United States, and especially applicable for New York growers with cucurbits. And so knowing how C. setosa may be able to impact these field dynamics is very important, especially for things like integrated pest management, in which pheromone disruption is a huge biological control that is being used. But it also opens up a great new field of looking at how parasitism is impacting chemical communication in insect species, especially since parasitoids are becoming more diverse and more frequent as biodiversity increases, and more insects are using chemical cues and chemical communication as a main way of communicating between each other. And so this will open up a lot of really cool, interesting new studies. Lastly, I'd like to thank the amazing mentorship of Dr. Jennifer Thaler and Matthew Barrett, and the entire group of the Toxic Plant Lab that made this summer so much fun. And lastly, Holly Myers for helping me with ImageJ, my friends and family at the University of South Florida, and all the funding from NSF, SARE, USDA, BTI, Cornell. Thank you so much, and I hope everybody has a great day.